Hi, welcome back. In the previous two sections, we have shown you how to extract the information from the drive dataset into individual files based on the cell lines. Now, in this exercise, what we are going to do is we are going to merge all those data into a matrix which consists of only the data about pancreatic cancers. Okay, so here is a data set that we have extracted previously. You can see that there is individual files consist of the data based on the cell lines. Now what we are going to do is we are going to select those cell lines that are actually pancreatic cancers and then compile it into a matrix format. So how do we know that which cell lines is actually from what type of tumors? In order to find out that, you can actually go back to the supplementary figure from the paper or the supplementary data under the supplementary data tool. Okay, if you open it, you can see that they will give you a range of annotation. For example, this is the cell lines, and this is where the primary sites belongs, and this is the classification of the tumors by the pathologist, for example. So what we want to do here is basically we are going to identify what are the cell lines that are actually originate from the pancreas or pancreatic cancer. So we will filter it by pancreas. Now here you will get a list of the cells, a list of these cell lines which is considered as okay pancreatic cancer. So once you have identified which cell lines are belongs to the pancreatic cancer, the next thing is we are going to use the Delimit software again to basically compile the individual file into one single matrix. So let's open the Delimit program here. Okay, so now in here, basically we want to join, okay, all the individual file into a single matrix table. So now, so in the join files category, we will select horizontally because we want to join it horizontally, and then we can select what are the cells type that we want to join. So these are the individual files. So now what we need to do is basically we need to select the cell lines that are pancreatic cancer. For example, okay, BXPC3 um, is one of it, and then we also have CF PAC1. For example, you can go down the list and then continue to select all the cell lines that this belongs to the pancreas. In this exercise here, I'm just going to um, show you okay how it would looks like, okay, to join a few files so then you can try it out later on with the whole data set. So in this case I'm just going to join, okay, three files here. You will see that, oops, sorry, you will see that, okay, and BXPC3. Okay, so you will see that, okay, three files was being selected. Now you can also select, okay, whether you want to join all the columns. Basically, if you remember from the last exercise, each of the extracted individual file actually consists of some of the common column. For example, okay, in BXPC3, if we open it up, so you will see that the first two column is actually universal across all the cell lines. So we actually don't need to include that. So what I'm going to do is basically I will ask the Delimit software not to join the first two columns except for the first file. Okay, so that all the data from different file will actually match it to the individual rows and also the variables. Now, delimitation rules, you have to select basically comma, because when we export it out, it is actually a CSV file or comma delimited file format. So it will be by comma, and then result file name. Then you can name any file name that you want. Okay, in this case here, I name it as test, and then after that, I just need to press the join uh, button. So you can see the progress bar here. This is the output of how it will looks like. Again, you will see that the first two column is universal across the CF pack file. This is match it to the DANG cell lines file, and this is match it to the BXPC three files. And again, if you go to the the Excel file format, test whether it is here, you can then actually open it up, and then you in the Excel file. So once this is completed, so if you complete the whole selection of the file, you will actually get something that okay uh, look like this. So this is the one that I have done previously, basically just to show you how it would look like after the compilation. Okay, you will look at these are all the pancreatic cancer cells in the particular in the matrix table, but this is not end yet. We still have a lot of work to do on it. So, and in the next step, what we are going to do is basically to match each of the shRNA back to the gene level. Okay, so now that you have the matrix table, so here what we are going to do is we are going to match the individual shRNA into a particular gene. So if you remember correctly, the drive project actually is a shRNA library screen, and each gene was actually targeted by multiple shRNA. So in order to identify which shRNA targeting which gene, so we have to go back to the supplementary table again, okay, or the raw data set where there's this file called the shRNA gene map. So if you open up the shRNA gene map, so what you will see is that okay, this is the sequence, and then this is the gene symbol basically, and then you also have the entree ID to refer to the gene ID. So what we want to do now is basically is to compile this information, okay, into this matrix table. And the way to actually do it is that we can use Excel, okay, there's something what we call the VLOOKUP format. So we will create a few extra column here. So we are going to copy the heading, okay, from the gene map. So you have the heading here. Now, so what we are going to do is basically now, okay, we have to, so we will actually match the sequence back to the gene symbol. And the way to do it is that you can use a VLOOKUP function. You can learn about it from online, how this is basically done. So first, it will tell you is that the VLOOKUP, the first value will be the lookup value. I'm going to match this, okay, with the table data from here, and then return the value from column number 2, which is a gene symbol, and then it must be a exact match. So that's why I will put false. Okay, so it's a very powerful well, it is actually a very it is actually a very powerful command, okay, in Excel that you can actually use it for many things. Now you see that that's coming up. So okay, so you can actually check whether this is correct and go back here, and then you can search the sequence. So whether okay, so this is ALPPL2, so same as here. Okay, so you can repeat this basically to match with the okay on-trace ID. Now again, the lookup value will be here. The map is the same, but then now you want to return the number from column number 3, and then here you go. You will get that. Okay. So now all you need to do is basically you copy um, the whole formula through the to the end of data set, and then it will then automatically okay match the sequence okay here to the gene set, while to the gene symbol that is on the gene map table. Okay, so it takes a while, and here you go. So now it is all properly matched. Okay, now we have created the matrix table. Okay, so this will be considered our raw, the original matrix table. So we can refer to this later on if we want to make any other changes. Now, without disturbing the original data, I'm going to create, okay, and not basically create another copy. So if there's anything, so that I can always come back to this page here, okay, the original matrix table that we have created. So I will create a copy of this into a different, into a separate um, Excel sheet. Okay, so it will take a while because the file is quite big. All right, and here you go. So I would then name this as original, and then this would be okay. But it's a step that we need to do next is basically now is to remove some of the sequence that do not match to any of the genes. So now you will notice that okay, if we filter it by gene symbol, basically you will see that okay, there are some there are actually some genes which is actually not matched to the sequence. So you can see that some of these like the okay 00, these are basically some of the control shRNA that they use. So what we are going to do is we are going to remove this because in the following step, so the system would not be able to recognize this gene symbol. Okay, so we are going to remove all this data, okay, just basically by deleting it. Okay, again, it will take some time because your data set is quite huge. Okay, once you finish doing that, there's a few more things that we can do to clean it up. For example, okay, like the column on experiment and pool. Well, this information is basically not required for our analysis. We can actually delete those columns so that we will know that, okay, to basically make the size of the file smaller and also much easier for you to do the downstream analysis. You can do this for each of the cell lines in the matrix file. Now, the important thing that I want to show you is basically now there are some cell lines which do not have a complete data set. Okay, for example, okay, I can show you is that even in the paper it did mention that okay, some of the cell lines uh, the control was actually quite low now. Like for example, this one here, the Maya Paka 2, the plasmid is a control. This is after the set incubation period, and you can see that the control actually somehow there's a technical failure in the experiment. So the and the way they So after some of the cleanup, so the data should look like this. Basically, each of the columns, you have two columns here, the plasmid control and also the sample count, and then